Welcome everybody. Thanks for coming to the monthly meeting of the New York City Neighborhood Naturalist Group, which is part of the New York City EcoFlora project. My name is Lydia Paradiso, and I'm a doctoral student at the New York Botanical Garden and a graduate fellow with the New York City EcoFlora project. NYBG created the New York City EcoFlora project back in 2017 with a grant from the Institute of Museum and Library Services. It is a community science project with three main goals: to investigate urban ecosystems and urbanization, to support open source biodiversity data, and to increase understanding and appreciation of plant life. The program has since expanded to 4 partner gardens throughout the US, Chicago Botanic Garden, Denver Botanic Gardens, Desert Botanical Garden in Arizona, and Marie Selby Botanic Gardens in Sarasota, Florida. To date, we have had nearly 28,000 observers make over almost 800,000 observations of plants and their biotic relationships in New York City. And you can explore those observations in our project page on iNaturalist, which I'll post in the chat in just a minute. Each month, we also have an EcoQuest challenge, which encourages New Yorkers to observe a particular species or genus and their ecological partners, and then post them on iNaturalist as well. The EcoQuest for July is entitled Juicy July, which is focused on the genus Rubus, which includes blackberries and raspberries. So far, we've had 8 different species observed and potentially sampled as well. So I will put in the chat right now, we've got a couple links to, sorry, I have it minimized for here, okay. We've got our website on the NYBG website. We've got our iNaturalist project for the overall EcoFlora project, and then the iNaturalist project for this month's EcoQuest. So feel free to check those out. There's tons and tons of observations. And if you're not already a member of the project, to consider joining on there. So before we get into our talk tonight, I want to introduce our new EcoFlora project manager, who's going to be working with me, Tomi Barrett. Tomi is passionate about the confluence of people and the more than human world. She has worked in institutions like Denver Museum of Nature and Science, where she worked in the arachnology department as an assistant curator and spider educator. She's also worked in various gardens as an environmental educator, and most recently in California as a naturalist at an outdoor school. She's excited to be working with NYBG on this project and to help fulfill its mission of connecting people to the earth. It's been really great to have Tomi on board so far, and we're looking forward to holding some in-person plant walks and other events throughout the city, as well as some more continuing with these virtual seminars. If you are not already on our EcoFlora mailing list, you can sign up by sending an email to ecoflora@nybg.org, and so you can stay updated on these events as they're going. So I'm going to turn it over to Tomi.
Hi, everybody. It's great to be here for my first seminar. I'm really excited about everything that we're going to learn about in just a moment. I would like to start off by doing a land acknowledgement. So I'd like to begin by recognizing the original people of this land, the Lenape people, as well as their deep connection to the Lenape-Hoking homeland, which is where we are right now. As an organization that's dedicated to advocating for the plant world, I think we must also acknowledge those who are original stewards of the land. And I believe it's really important to acknowledge the settler colonial genocide that was perpetrated against the Lenape people, which is a community that still today is continuing to resist erasure. So without further ado, I would like to introduce our speaker tonight, Allyson. Allyson is an environmental scientist and urban ecologist who studies how trees and other plants grow in a built environment. Her research helped us understand how to help trees grow better in less than ideal conditions so that we can create greener places for people to live, work, and play. She has studied stormwater management systems, post-industrial sites, highway roadsides, hurricane impacts on trees, and street trees. She received her BS from, is it Susquehanna University?
Susquehanna.
Susquehanna University, and her Ph.D. from Rutgers University. She is currently an adjunct researcher for both Temple University and the University of Florida. And prior to her current positions, Allyson worked at the Watershed nonprofit in Northern New Jersey and New Jersey State Forestry Services and the Morton Arboretum. In her spare time, she enjoys gardening, cooking, knitting, hiking, board games, and Dungeons and Dragons. In tonight's talk, Allyson will discuss her work on plant biodiversity and succession at Liberty State Park in New Jersey, a fascinating ecological case study about what happens when a former rail yard and industrial site are abandoned and left for nature to recover. She will also share the history of Liberty State Park and some of the ecological lessons learned from its interior forest gleaned from over a decade of research. So please feel free to enter any questions or comments into the chat as we go. At the conclusion of the talk, we will have we will go through as many questions as we are able to. If you have a question or comment that you would like to ask verbally at the end of the talk, you can use the raise hand feature or note in the chat and you will have the opportunity to do so. And in fact, we encourage it. Also, if you're unable to stay for the whole talk or would like to share with others, a recording will be posted on our YouTube page where you can find recordings of other recent talks. And then also on that YouTube page, feel free to like and subscribe to keep updated or just see all of the other talks that we've done and are going to do into the future. All right, Allyson, go ahead and take it away.
Awesome. Thank you so much for that introduction, Tomi. If you'll give me a few moments to get my screen and my Zoom, my screens oriented. Are you guys seeing the screen okay? Or my presentation all right? Hopefully, hang on, I can't see anybody. We're good to go?
Yes.
Okay,
Good.
awesome. Fantastic. So hello and good evening and welcome to my talk about plant biodiversity at Liberty State Park in Jersey City, New Jersey. Come on. Slide advanced 30 seconds ago. There we go. All right. So this talk should be about 45 minutes and is going to be split into 4 parts. Like Tomi mentioned, feel free to drop your questions in the chat as we go along, and we'll definitely have time to answer some questions at the end. So let's start off with some introductions. As Toby mentioned, I am an environmental and urban forestry scientist who studies plants that grow in generally pretty tough environments. This is a photo of me at a previous job in Illinois. I'm standing in front of a pile of treated sewage sludge that was used to improve highway soils prior to tree planting. I'm currently part-time research staff, a remote staff member at 2 different universities. At Temple University, I study plants that grow in stormwater bioretention basins next to highways. And at the University of Florida, I'm studying how hurricanes impact urban trees. My main role at these institutions is to analyze data that's been collected by my collaborators and use it to write and publish scientific papers. The research that I'm talking about today was primarily conducted at Rutgers University. Rutgers is where I went to graduate school and also spent time as a postdoc, which is a special type of research assistant. And as a disclaimer, all opinions in this presentation are my own and do not necessarily reflect those of my employers. As Tomi also mentioned, the research that I'm sharing today was conducted on the ancestral lands of the Munsee Lenape. And as a side note, if you're interested in learning more about the Lenape people, I highly recommend checking out the Lenape Center. It's a fantastic cultural institution that's based in New York City, and they are currently working to build a cultural center and meeting space to serve the Lenape diaspora. There was a recent set of exhibits and programs at the Brooklyn Public Library. I have a link down in the corner of this page And they had recorded all of their talks. So when you're done watching all the cool YouTube videos from this series, you should check out those that were put on by the Lenape Center and the Brooklyn Public Library. So I was born and raised in the weird and I believe wonderful state of New Jersey. I grew up staring at post-industrial landscapes in northern New Jersey on weekend car rides, visiting my grandparents up in Bergen County. I was always really interested in the plants that were growing in these kind of surprising places between buildings, and railroads, and power lines, like you can see in this photo. Like, it's kind of a little surprising, like, why are plants growing here? As an adult, I had the privilege to actually study plants in that landscape as part of my PhD research. In graduate school, my research built on the work of Dr. Frank Gallagher at Rutgers, New Brunswick. He really spearheaded a lot of the early and ongoing research in the interior of Liberty State Park, which, as a side note, you might hear me call or refer to it as LSP, because I'm a science person and need to abbreviate everything. So I might call it Liberty State Park, might refer to it as LSP. They're the same thing. I also want to acknowledge that my research would not have happened without the wonderful bunch of people in this photo and listed on this slide. Science is absolutely a group effort, as the EcoFlora project demonstrates. So again, the research I'll be sharing here today wouldn't be possible without these folks. After my PhD, I continued to work or conduct research related to Liberty State Park with Dr. Klaus Hutzapfel's lab at Rutgers Newark. He led the 8-year data collection study that led to the biodiversity research I'll be talking about later in this presentation. Again, that research was also a group effort involving many members of the Hutzapfel lab. So to fully appreciate the contemporary research that's been going on at Liberty State Park, I need to walk you through the history of this site. That history begins before the arrival of European settlers. That land is, the land that is currently Liberty State Park actually wasn't land at all over 200 plus years ago. This land was a tidal marsh. It was used by the Lenape people for gathering food from a region called the Communipaw. So on this map that you see on your screen, Communipaw's kind of original outline is shown on the map. I can make my little, try my little laser pointer. So this line represents the original boundary of land in what is now Jersey City. And then this bright blue line is the current waterfront line that was created and extending the land of Jersey City into New York Bay. So as colonial influence spread and grew, enforcing native peoples further west, this land for a time was known as New Netherland, sort of an extension of New Amsterdam across the river. Following a land dispute over the waterfront with the city of New York in the early 1800s, Jersey City officially became a part of the state of New Jersey and became Jersey City in 1820. So, and you can see from this drawing here how during this time period, the city was very much a bustling port and industrial center. So around this time period, again, the mid-1800s, the railroad industry wanted easier access to the deeper waters of New York Harbor and access to those ships that were bringing in commerce. So they started using construction debris, dredge spoils, garbage, old barges, and other sorts of unwanted materials to destroy the marsh or to bury it and create dry land. So this photo, let me get my laser pointer back up. So this photo shows a soil profile from the center of Liberty State Park. The ruler is metric. It's about 3.5 feet deep. What you're seeing in this photo is here's the grass on the surface. And if we go to the bottom of the pit, we have this clay or very gray, muddy looking material. This may have been dredge spoils or perhaps old marsh soil. On top of it is a very thick layer of sand. These are some white things or seashells. So this is probably other dredge material that was moved from another site and starting to build up the land base. And then this next layer is a lot of gravel and other industrial materials of unknown origin in this very dark layer. And then on top of that, we have some other layers of things like cinder ash. There's a lot of slag material, which is a byproduct of ore smelting, and then a much thinner like soil, what we would call an A horizon, and maybe a little bit of organic matter on the surface. For contrast, this photo shows what a naturally formed soil profile can look like. You can see there's a much gentler gradient of color and texture as compared to these very abrupt brown boundaries of extremely distinct and disjunct material that have formed in this anthropogenic or man-made soil. So this is one of several interesting features of the park. So zooming back out a little bit. So once dry land was created, moving out into the harbor, this land was used for about 100 years as a rail yard and the location of several industrial facilities, including a chrome plating factory. So in this photo, I should just leave this laser pointer up. So in this photo, you can see the Central Railroad of New Jersey Terminal. This historic building is still at Liberty State Park. You can go and visit it. It's very beautiful. It has actually a very special place to me, or place and connection to me personally, since my great-grandparents and great-great-grandparents from Italy came through Ellis Island and likely went through this terminal to take a train here and start their new life from there and start their new life in America. So you can see behind it the numerous both passenger railroad lines, and then on this side, which is where our study area is located, is where all of the freight trains would come in and then either pick up material or drop off goods at these docks in the harbor. So unfortunately, fortunately or unfortunately, I guess, the property was abandoned in 1969 when the Central New Jersey Railroad went bankrupt. Their operations were consolidated and relocated. At the same time, industry in this region was in decline. And so this land was abandoned, and what was left behind was an industrial wasteland, which was a very unfortunate eyesore as the now backdrop of the Statue of Liberty, this iconic site. As a fun TV or movie trivia side note, during this time period, the land that would become Liberty State Park was featured in a classic scene from the movie The Godfather. Feel free to drop the famous line from this particular scene in the chat if you know what part of the movie this is from. So lucky for us, several local activists from Jersey City saw the state of the land behind Liberty State Park, and they saw opportunity. They saw great potential in the mess that was left behind. Those activists include Morris Pezin, Audrey Zapp, Theodore Conrad, and J. Owen Grundy, who worked together to lead a movement to purchase this land, or have the state purchase this land and convert it into the park that's there today. Liberty State Park's grand opening was in 1976, and it was kind of included as part of America's bicentennial celebration. Oh, and I forgot to label, but in the two photos I have, the gentleman is Morris Pezin and the lady is Audrey Zapp. And from there, we will jump to the present. Today, Liberty State Park encompasses over 1,200 acres, and it includes a beautiful waterfront, a historic railroad terminal, there's playgrounds, picnic areas, walking paths, boat launches. There's a lot, beautiful scenic views of the New York City skyline. There's a lot of just really wonderful stuff at this park. If you can't tell, I'm a really big fan. But actually, before the public was able to access this park, its soil pollution had to be managed. This pollution included both heavy metals and polycyclic aromatic hydrocarbons, or PAHs for short. So the outer perimeter of the park, which kind of makes like a U or like a backwards C shape. It kind of goes from Caven Point here to the southern boundary, to the entire waterfront, to the terminals here, over by the marina, and Liberty Science Centers right here. So this whole area was treated with a few different remediation techniques. That includes capping. There was a treatment wetland installed, as well as groundwater pumps. However, due to funding limitations, the park's interior area could not be treated. So that's this area highlighted in yellow here in the middle. That's kind of this much darker shade of green. So this area was fenced off, and for the most part, kind of left alone, and nature was left to take its course in the middle of the park. Move my screen. So, while unfortunate in many regards, not treating the park's interior led to the development of a unique ecosystem that naturally developed with minimal human intervention on contaminated and fill soil. There's a decent amount of other research out there about the development of ecosystems on post-industrial sites. But these research projects often focus on the very early years, maybe the first few years, maybe up to a decade of plant community development on abandoned industrial sites. But what's really special about Liberty State Park is that we have an opportunity to study a novel ecosystem that has been growing and developing for decades. So around the time when research really got going, or I should say academic research, there were other kind of surveys and studies done during the creation of the park. But academic research really got going when the ecosystem was probably between 30 and 40 years old, which is a really special opportunity. And that brings us into our next section about the academic research that's been conducted at the site. But first, I'm going to take a swig of tea. So, as I mentioned, the interior soil is contaminated with several heavy metals and polycyclic aromatic hydrocarbons and a few other petroleum products. So for most of the rest of this talk, I'm going to be focusing on the heavy metal contamination, since that's been the focus of most of the plant-based research at the site. The main reason for that is the kind of the higher levels of heavy metals. Most of these metals exceed either residential or ecological safety standards, and they're pretty ubiquitous and found throughout the entire interior. So the heavy metals that are the focus of our research include arsenic, copper, chromium, lead, and zinc. So interestingly, while there are heavy metals throughout the site, some areas of the interior have higher concentrations of heavy metals than others. So this gradient creates a really convenient natural experiment and lets us study how plants respond to varying levels of heavy metals within the same general area. So for example, on this map, which shows lead concentrations, it's been observed that these darker lines, the darker red lines, indicate higher concentrations of lead, and the lighter lines indicate lower levels. So each of those 5 metals I described kind of has their own unique gradient and own version of this map, which again highlight the very, what we would say, heterogeneous nature of pollution within the interior. So in Frank Gallagher's early research at Liberty State Park, he found that the most informative way to study all of these 5 metals was to actually combine their concentrations into a single score, which he calls the total metal load. We also refer to this as TML. Again, a few more acronyms. Total metal load is what we call a rank sum index of the 5 metals. I'm not really going to delve into the math on this, but what's important for you to know is that a low total metal load score means lower overall heavy metal concentration, and a high TML score is associated with higher concentrations of heavy metals. And our scale ranges from 1 to 5. So this index was based on 38 study plots within the LSP interior, and it is unique to this particular site. The kind of one drawback of this method is it doesn't really facilitate easy comparison with other contaminated sites, which may have a different cocktail of heavy metals or pollution. This index is unique to the park. I'm going to pause for a brief science terminology note and reminder. In this presentation, you'll hear me refer to ecological or plant communities. And in this context, a community is a collection of species within a given place. So in ecology, this might be all types of species, like in this diagram here, of animals and fungi and plants and insects and microbes. Or we can also refer to a community of a more specific group, like a community of plants and a collection of plant species in a place. Another interesting feature of the interior is that it contains several different plant communities. Each color on this map indicates a different community type. And while this map shows kind of broad categories like forest or grassland, each of these categories can be split into more refined groups. So for example, our shrublands include things like a maritime shrubland and an early successional shrubland. But for simplicity's sake, we're going to keep with this higher grouping today. The plant community includes both upland environments as well as wetland environments. Again, today I'll be focusing on the upland, the 4 upland habitat types: the grassland, the forb herb, the shrubland, and the forest. Your previous speaker, Dr. Monica Palta, she actually did a bit of research on the wetlands growing in this site. So if you want to learn more about the wetlands here, I would refer you back to her research. I will be kind of skipping over that today. So at LSP, our early successional forest community includes fast-growing pioneer species like gray birch, quaking aspen, and cottonwood. These are species that love sunlight and are really not picky about the soil conditions they are growing in. Our shrub community includes species like winged sumac, northern bayberry, and autumn olive, among several others. LSP has 2 types of meadows. One is dominated by grasses such as calamagrostis, red fescue, and purple top. And then our other meadow type is dominated by other forbs or herbaceous species like goldenrod and mugwort and other asters. So Frank Gallagher and other LSP researchers have kind of taken advantage of both the gradient of heavy metals and the mosaic of vegetation communities and kind of use this to kind of create experiments and serve as the foundation for several different research projects that I'm going to give like a very brief and quick overview of. So through some of Frank's early research and research in his PhD, we've learned that within the interior, both plant productivity and gray birch growth rates decline as heavy metal concentrations increase. So you can think of plant productivity as the overall amount of living green vegetation in an area. So more metals, slower growing plants, and kind of fewer or less abundant plants overall. Frank also observed that there tends to be more early successional forest habitats in our high total metal load soils. And interestingly, those forest habitats established earlier on our more contaminated soils, as Frank discovered looking at historic aerial photos. By contrast, our low total metal load plots tend to be occupied by meadow habitats instead of forests. A study of both house wrens and robins showed that while these birds, not too surprisingly, these birds did tend to have higher concentrations of heavy metals in both their feathers and their poop, which means that they were being exposed to the contamination from the soil. However, when Liberty State Park birds were compared to birds from non-contaminated sites elsewhere in New Jersey, they found no difference in the size and survival of their chicks. And so that's good news. So there is some evidence of exposure. However, this exposure does not seem to be having substantial impacts on these two bird species. Another researcher, Yu Quinn, and their team observed that some plant species, such as gray birch, winged sumac, and mugwort, can accumulate some of the heavy metals from the soil into their stems and leaves. They also, these species also accumulate metals into their root tissue as well. So we think this movement of metals from the soil to above ground and then returning to the soil as plants drop their leaves or die and return their biomass to the soil, we think this might actually be playing a role in helping to stabilize the long-term metal concentrations in the soil. So this is a process referred to as phytostabilization. It means that an ecosystem that's established on a polluted site can keep the soil or keep the pollution in place, preventing its spread. So that means that when metals or other pollutants get absorbed and bound into plant tissue, those metals are prevented from getting washed off into streams, getting blown away, or leaching into groundwater. And while not appropriate for every site, phytostabilization is a reasonably affordable method for containing pollution under appropriate conditions. So phytostabilization is a little in contrast to phytoextraction, which is a situation where plants sort of act like a vacuum cleaner to remove metals from the soil, and then the plant material gets removed from the site. And the premise is that over time, this sort of, if you will, plant vacuum action should reduce the long-term concentrations of metals over time or other pollution. The drawback is that this practice can be really challenging to implement and needs very particular conditions to work efficiently. And it's usually pretty expensive because at the end of the day, you end up with a bunch of plant material that's contaminated and you need to find something to put it safely to still deal with that contamination. So as part of my PhD research, I did a comparison of soil samples from the forested areas of the interior based on soil samples that had been collected over the course of 20 years by different groups. And my analysis showed that soil metals within the forest have stayed fairly consistent over time, which is kind of exciting because it suggests that the interior's forests are helping to contribute to the phytostabilization of the soil's heavy metals. So the heavy metal contamination has kind of stayed the same over time, and it means that that pollution hasn't gone elsewhere to spread and impact other places. So if soil metal levels are staying consistent over time, this raises the question of, can forest succession occur on this site if the kind of metal stresses more or less sort of stay the same over time? Traditionally, in the northeastern United States, plant communities change over time, kind of following a general pattern. We have start off with our herbaceous species that get replaced with shrubs, and then light-loving pioneer tree species move in. And then later, more shade-tolerant species establish until the forest consists of what is often called a climax community. Though granted, this is a very general pattern in plant community development, and there are many exceptions in many different places. But for the most part, we kind of expect ultimately abandoned land eventually grows into a forest with large mature hardwoods, and some softwood species as well. So, but Liberty State Park isn't exactly your average northeastern US natural environment. It's a man-made site, so with a good amount of pollution. So it's possible that soil conditions and other factors may contribute to favoring early successional species and kind of prevent the establishment of mid and late successional species like oaks and maples and hickories and other hardwoods. So to investigate potential successional patterns and changes in biodiversity over time, Klaus Hitzapfel set up an 8-year study within the interior. So in 2008 and 2016, he inventoried the type and amount of plant species in 27 permanent plots. And those are the little dots shown on the map in this slide. So after Klaus and his team collected data, my role in the project was to take their data and try to make sense of it all and see if we could answer some of the questions we had. Klaus's observations were not the first vegetation surveys conducted at the park. Several others have been conducted over time. One that I wanted to highlight that I thought was really interesting came from an article in Bartonia by Carl Anderson, and what I believe was just a group of volunteer botanists who surveyed Liberty State Park in the 1980s. And it's a really interesting snapshot of the plant community of the entire park, both the interior and the publicly accessible portion. Unfortunately, though, because their sampling methodology was so different than Klaus's approach, I couldn't quantitatively integrate their data into our study, but it did provide really helpful context and background information for our interpretation of our results. So It kind of reminds me a little bit actually of the EcoFlora project, and it sort of demonstrates how documenting plant species and plant communities is really important for informing our long-term regional understanding of biodiversity patterns in urban settings. So for our study, at each plot, that was each point on the map, Klaus and his team would set up a plot that was a 5-meter diameter circle. They would identify all of the species inside of the plot, and then they would estimate the percentage of the plot occupied by each species. And this metric is called percent cover. They also collected soil data for each plot as well. The plots were spread across those 4 upland habitat types I mentioned earlier: our grassland, our forb meadows, our shrubland, and the early successional forest. So one of the first observations from this data set showed that from 2008 to 2016, the number of plant species increased across all 4 habitat types, though the largest increase in species occurred in the shrublands and the forest, or forests. There's a few forest patches. So this graph shows how percent cover of each kind of category of plants, trees, shrubs, vines, and forbs, and grasses, changed from 2008 to 2016. Across the entire site and the 4 habitats. What we noticed was that most of the new species that were coming into these plots were primarily vine and shrub species. So that's the kind of light blue and the light green bars on this graph. This next chart shows the relationship between total metal load and the change in species from 2016 to 2008. So across the bottom of the graph, higher numbers mean there's more metal pollution in the soil. And then as you move vertically on the graph, that means that a plot gained more species over time. And what we observed was that our lower total metal load plots tended to gain more species during the 8-year study period compared to the high metal concentration sites, which gained fewer species. Notably, the percent cover of introduced and native species really didn't change very substantially during this time period. So we thought that was kind of interesting that our native to non-native ratio, at least cover-wise, was pretty consistent. The 2016 survey observed 4 new tree species. Though interesting, none are considered true climax forest species. The red oak, red maple, sycamore, and hawthorn are all pretty flexible in their light adaptation. They can thrive in high amounts of sunlight or a bit lower amounts of sunlight. And so they can they're not really indicative of like a low, middle, an early, middle, or late successional status. So it's interesting that we see new tree species moving into the site, but it's not really giving us a clear idea of the kind of future trajectory of the forest just yet. So we have a few possible explanations about why plant diversity was increasing during this time period. It's possible that soil conditions have become more favorable as soil organic matter has accumulated over time. So as plants grow and then return their biomass and dead parts to the soil eventually becomes more enriched with organic matter and nutrients, and its condition improves over time. And that may favor the establishment of new plant species as the soil condition improves. One important thing to note, an important event that happened between the first and second, or Klaus's first and second plant survey, was that Hurricane Sandy stopped by and unfortunately inundated most of Liberty State Park with anywhere from 1 to 3 feet of brackish bay water. So unfortunately, this did have a pretty substantial impact on the interior. We assume that there were species that were probably exposed to salt water that died because of that exposure as well. There were areas where quite a few trees have fallen over either from the winds or the flooding. And so these dead trees plus fallen over plants probably created or were creating new gaps in the forest canopy and perhaps making room for new plant species to move in. And our third potential or idea or potential explanation is that it's just taken time for seeds to reach Liberty State Park. Though perhaps with new greening efforts in the region, maybe seed availability might be increasing over time. So on this map here, Liberty State Park is highlighted in yellow, and you can see that its entire region is quite densely urbanized. So there's not a lot of other green spaces to provide sources and seeds of new plant species. And so that may also explain why changes in the plant community at Liberty State Park have happened rather slowly. It's probably just takes a long time for a seed to get from one natural area that's nowhere near Liberty State Park to that interior and to establish. So again, I want to emphasize these are all hypotheses and ideas that might explain the data that I was just showing you. We haven't had the opportunity yet to test them. So that was a very quick and brief overview of some of the plant research at Liberty State Park. I assure you it is the tip of the iceberg. But to briefly summarize, what we have learned is that soil metal contamination affects plant growth. We have learned that it affects the type of plants that grow in a given location, and it affects how the plant community changes over time. We've also learned that the interior's plant community is still continuing to develop and change. And there's probably several different factors that are contributing to the nature of those changes. So what might we expect for the future of Liberty State Park? So from the perspective of the plant community, I think, I hate to say it, but I think we're still kind of waiting to see what might come next for it. It seems equally plausible that plant communities could stay fairly stable over time. So we would see kind of a similar composition and group of species that's there now may persist into the future in this what's described as arrested succession pattern. Or it's also possible that rather than seeing a traditional succession trajectory, we may see something in the middle where the plant community continues to change, but it develops into a more novel group of species that's perhaps a little unique from other forests in the region, but is still different from what's growing there now. There's also a lot of other exciting research that's been conducted at the interior of the park. I want to give a shout out to and highlight Dr. Kathleen Farley's recently completed Ph.D. research about woodcock usage of post-industrial sites like Liberty State Park, as well as research by Dr. Jen Crummins, Nina Goody, and Diane Hagman, who have been studying the relationship between soil mycorrhizae, which are symbiotic fungi that grow with plants, and how those fungi might be helping and facilitating the growth of plants despite exposure to pollution like the heavy metals and the PAHs. So they have a fairly new paper out in environmental microbiology. It came out this year, and it's got some really, really interesting research in there. So also recommend checking out their work as well. And I would be remiss to not acknowledge that Liberty State Park has also been in the news recently. Our state legislature has passed a bill that opens the park to commercial development. And this is a deeply contentious issue, especially with people who live around and use the park regularly. There are a lot of people who express concern that the bill is lacking limits on the extent of large-scale projects that could possibly disrupt the park's character and accessibility. There's a lot of great articles online that explore this issue in more depth, and there are a lot of groups out there who are, or several groups out there who are working to try to promote the best interests of the park and the people who use it. So I recommend learning more about that if that's a topic you're also interested in. So there are over 450,000 sites like Liberty State Park across the United States. They're referred to as brownfields, and they are properties that are difficult to redevelop or reuse because of the presence or potential presence of pollution. And again, that 450,000 number is just in the United States. That number is probably much, much greater globally. And while I wish we could remediate the pollution at each and every one of those sites, unfortunately, that's more of a wish than an achievable goal. But what's maybe on the bright side is that Liberty State Park has shown us that in some places and contexts, phytostabilization can be an effective management approach to reduce and limit the spread of pollution from a contaminated site. It's not a solution for every location, but it can be a viable option. And also, I think I kind of want to end on the note that I think it's just worth admiring as well what nature can do when we leave pretty serious messes behind. Again, it'd be great if we didn't do that, or we could get rid of those messes. But in lieu of that, nature's pretty resilient and can do some great and really fascinating things. So I hope I've inspired you today to take a bit of a closer look at the forgotten parts of our urban landscapes and appreciate what you do find there. So thank you so much for your time. I really appreciate it. And I would be happy to take your questions. And
Awesome.
let me know if it's easier. I can unshare the screen, or I can leave it up if people have questions about the presentation. Also, we can start off. Did anyone get the movie quote?
Looks like somebody answered that in the chat.
What did we get?
I can't say if they're right or not. So maybe we'd have to have a consensus. But Jill Bauer said, leave the gun, I'll take the cannoli, leave the gun, take the cannoli. Yes,
that is the that scene. So shot at what would become Liberty State Park. So it's always there's a fun bit of trivia Frank Gallagher taught me that I enjoy also sharing because it's pretty funny. And it's a great scene. And who doesn't love cannolis? So
I actually haven't seen that movie, Godfather. I know it's like
well, now you have an environmental reason to.
Now it's related to work.
Yeah.
Okay, so we have several questions in the comments. Some of them it looks like you answered. You have some congratulations as well.
Oh,
so let's see. E. Hoffman I guess for a quick clarification, E. Hoffman was wondering, was all of Liberty Island created by landfill?
So the land, that is a good question. Let me hang on. Let me go back to that map I had before.
Well, and actually, while you're doing that, I just want to make a comment about landfill because it's crazy to be. I remember learning like how much of Manhattan, for example, is like the bottom parts of Manhattan that were created, like how much land is created by landfill, that you're just standing on something you assume is the natural dirt that was there, that this was, you know, the land, it's, you know, continental parts of land, that it's both like really amazing that we can create more land, but also right at what cost? What are we kind of the, you know, trade-offs for that kind of thing?
Absolutely, right? Because, you know, look what got under 3 feet of seawater when it could have been a marsh, right? So yeah, that's the price we kind of pay now. So there's a question about if Liberty Island was made of fill material. So I assume, so what I do know and I'm very confident of is that the kind of part of Liberty State Park that is attached to the Jersey City Peninsula, that one is completely fill. What is actually Liberty Island, where the Statue of Liberty is, I am not entirely sure if that was originally like a bit of an island that got bigger, or if it was completely man-made. I don't know, and that's really interesting. I truthfully know a lot less, even though they're like literally a boat ride away. I truthfully know a lot less about Liberty Island and Ellis Island, though I think you can kind of see from the map here, if you squint, this kind of little ellipse shape where Ellis Island is. On this historic map was a lot smaller than the current extent of Ellis Island. So Ellis Island was probably filled in quite a bit, but the Statue of Liberty's island, I truthfully don't know. I'll have to look that up.
Awesome. Sandy asked a question about the mycorrhizal communities, which you mentioned in your talk. Do you have anything else maybe to add or thoughts about the mycorrhizal communities there?
Sure, yeah. Again, that is very much Jennifer Crummon's wheelhouse, but from what I can remember about her research is that, so one interesting feature was that they found different mycorrhizal communities, so different types of mycorrhizal fungi are found in different, they differ also with the total metal load. So there seem to be, and that suggests there may be mycorrhizal fungi that are more tolerant of the metal conditions, and that their recent paper does strongly suggest that mycorrhizal associations make it easier for plants growing at LSP to grow despite some kind of less than ideal growing conditions. So that's about the extent of my knowledge on that subject, but that's a really, I think, important avenue of research to keep investigating. Mycorrhizal fungi are really fascinating and awesome. They're also a royal pain in the neck to study because root research is tough.
Next steps.
Yeah.
All right, I would like to ask a question of my own before we continue, and it was just, what surprised you about the research?
Oh man, what surprised me about the research? I think,
if anything.
Oh, there were, especially when you're doing, I think, observational research, there's so much that's surprising. I think it just surprises me, like, the soil is awful. like we'd be doing soil sampling and there's like lumps of coal, there's slag with like, you can see the rusted metal. It's a very sandy and granular, like it dries out really quickly. And it just, as I got to know the site more, it honestly felt more and more surprising that such a relatively like abundant ecosystems were still growing there anyways. It was just like, wow, these plants really don't care. Like, I can't get a tomato plant to grow in my backyard, but this birch tree in this horrific soil, it's fine. I think maybe that was maybe one of the more surprising things of just both how bad, but how not a big deal at the same time things are.
Yeah, I think one of the things that surprised me was that in the time of your study, you found an increase of species diversity, even though the plot had been abandoned for 40 years already, which is surprising. And then I think in your paper you mentioned, quoted somebody, or maybe mentioned it yourself, that usually plots of land or like anthropogenic landscapes plateau, species diversity plateaus over or after about 20 years. Do you have any idea why it might be different in this plot?
Sure. Yeah. So that one thing we did in the kind of research paper that accompanied the biodiversity data I shared was we qualitatively made comparisons to like abandoned farm fields, for example, which kind of typically reestablish into forests, barring unforeseen circumstances, like pretty quickly. You know, over the course of maybe 2-3 decades, you can get a forest back. I think a big factor is both the isolation of the site. Like, there's just not a lot of other plant species to contribute, minus like really what gets planted around the interior. And then again, the really kind of weird combination of stressful soil conditions, I think all has kind of helped led this site to develop at what we would consider a slow pace for our region.
Yeah, it's crazy. Like, where are these seeds even coming from that are establishing at this site? There's no seed bank, so it must be surrounding areas and roadsides and things like that.
Yeah.
So Jack asked a question: what does the research tell us about considerations for planting native plants when brownfields have undergone remediation?
Well, the research first off shows us that there are certainly native species that can thrive and do well in this environment. So it's certainly possible to get native species to establish, and here they have established on their own, and they, for the most part, kind of coexist with non-native species as well. So that's may just kind of be an interesting feature or factor of this very, novel soil appears to lead to novel plant communities as well. So native species establishment is certainly not impossible, but it's also possible for them to coexist with non-natives.
Thank you. Okay, Ken asked, you mentioned that phyto extraction is difficult and expensive because it involves the removal and disposal of plants that become loaded with heavy metals. If I understand what was said correctly, why do they need to be removed rather than left to decompose or regenerate? Can you speak more to this?
Sure, I can. Yeah, so the premise so the reason in phyto extraction that typically it's expected that at some point the plant material is removed is that the goal of phyto extraction is to decrease the amount of pollution in the soil. So that in order to do that, you need to remove the pollution like from the site completely. So the phytostabilization says, we're not interested in changing the kind of total amount of pollution within the given site. We just don't want it to spread further away. And so that's why in the phytostabilization case, approach, you kind of let the plants, what they were describing, let the plant material, it dies, it returns to the soil, and then you kind of create this cycle where metals or the pollution's either in the above ground part of the plant, or it's in the soil, and then goes to the roots and then goes back above, and you kind of get this loop going. That's the goal of stabilization. Whereas extraction wants to reduce the total amount of pollution at the site. So that's the main difference between, one main difference between the approaches is their kind of end goals. Does that hopefully help clarify?
And to follow up on that, what do they incinerate the material? Do they put it in some, like, because I guess you can't get rid of the metal, will still be there in some capacity, but I guess.
Yeah, so incineration is one management option, or just landfilling the plant material into more safer and contained landfill. That's another option. I think those are the two big ones that I can at least remember off the top of my head.
I also wonder about how these plants are creating a stabilization of the metals, but also I'd imagine that there's some dispersal of the metals happening. Do you know if any heavy metals are carried in pollen or seeds, or is it mainly just leaves and roots, plant body?
So in Yu Quinn's work, I know they looked at roots, stems, and leaves. They didn't look specifically at pollen or fruits. Very, very generally speaking, when plants are uptaking metals out of the soil, or in particularly heavy metals, plants try to, because fruit and pollen, those are reproductive structures, and reproductive structures are really important to plants. They, I think, generally speaking, plants try to minimize putting potentially damaging materials into those reproductive structures. So generally, fruits and pollens, I believe, tend to have lower metal concentrations compared to other plant parts. A lot of metals will end up, metals that are mobilized and able to move throughout the plant usually end up in the leaves, because that's where water is going as the plant is transpiring. So, I mean, it's certainly plausible that, yeah, if you're gonna have leaves getting blown here and there, you know, during autumn and such, but that movement is much reduced compared to a site that would have no plants on it at all.
Great. Right. Thank you. Okay, so Dave Taft asks, well, first he says, phenomenally interesting. Thank you. Then he says, wondering about the potential differences in several sites I know from what I understand of your site. In the sites I refer to, oh, I lost it. Oh, to invasive vines seem to predominate after just a short few years. Poison ivy also becomes very interesting and troublesome. You mentioned woody vines also. Most of the non-viney species you highlight seem to be natives, mugworts and olives excluded. Yeah. So is there a difference in this site than other maybe disturbed sites that you've seen in your life?
I'm glad you brought up the vine conversation. That was something else I think we touched upon in the paper we wrote. And your observations are is not you. Quite a few other researchers have documented that woody vine species and cover are maybe becoming a bit more abundant in our region for a variety of reasons. One of which is habitat fragmentation. Vines like to take advantage of most woody vines in our area like to take advantage of sunlight. And so I think, so when you, in other sites, right, where a forest is maybe gets a road put through it, or half of it gets turned into a shopping plaza, you have like new edges that form, and that enables like vines to move in, like, ooh, more sunlight and things to grow up. I'm just gonna mooch off this tree and make my way up it and establish So that is a common pattern that's observed. I don't know why I can't say that word today. Observed in a lot of northeastern forests. And at Liberty State Park, I think some of that increase in vines species is probably something similar. A lot of gaps in the forest canopy were created because of Hurricane Sandy. And so I think that did probably do some, the formation of those gaps, plus the fact that at this point in the forest's history, there's a pretty decent layer of organic matter has started to develop, that it was sort of like a prime opportunity for vine species to move in. So yeah, there are a few plots where it's just all vines, and those are not fun plots to visit. So I think it's actually not quite as different as some of the other sites that yeah, he was describing. So
Thank you, Allyson, so much for your talk and answering all of our questions. Thanks everyone for being here. Even though, you know, it's in New Jersey, it's really all
we're
the
just
same,
right
you know,
across the river.
So far.
and it's, you know, all the same, our geographic boundaries have little to do with, you know, the ecological, et cetera kind of things. Stay well, everybody, and hopefully we'll see you soon.
Thanks, everyone.
Allyson.
Take care.